Hello everyone, in this class we'll try to learn about the visual process which occurs in rods and cones. Receptors. Visual pathway starts from receptors that responds to electromagnetic stimulation. Each rod and cone is divided into an outer segment and inner segment that includes a nuclear region and a synaptic zone. The outer segments are modified cilia and are made up of regular stacks of flattened saccules or discs composed of membrane. These saccules and discs contain photosensitive compounds that react to light, initiating action potential in the visual pathways. The inner segments are rich in mitochondria. The rods are named for their thin and rod-like appearance of their outer segments, and cones generally have a thick inner segments and conical outer segments. So these are the structures of rod, which is a rod shaped, and cone, which is a cone shape. So these are the outer segments. These are the inner segments. Outer segments contains stacks of discs, and inner segments contains the nuclei. And usually the light falls. This is the direction how the light falls on the these visual pigments. Some of the difference between rods and cones. Rods, the outer segment is rod shaped with regular stacks of membrane disc, whereas in cone, outer segment is cone shaped. In rods, discs or saccules are separated from cell membrane, but in cones, discs are infoldings of the cell membrane. Rods has a photopigment called as rhodopsin, and it contains opsin, that is scotopsin, and vitamin C, whereas in cones you see three types of cone pigments: erythrolabe, red, cyanolabe, that is blue, and chlorolabe, that is green. Except in fovea, rod predominates everywhere in the retina, whereas the cones are there abundant in fovea. Rods helps in the dim light vision, whereas the cones, they help in bright light, color vision, and visual acuity. So there are about 120 million rods in retina. There are only 6 million cones in the retina. Photoreceptor mechanism. Visual or phototransduction is a process by which light energy is converted into receptor potential in visual receptors. Potential changes that initiate action potential in the retina are generated by the action of light on the photosensitive compounds in rods and cones. The conformational change which happens when light falls on these substances trigger a sequence of events that initiate generation of action potential. Ionic basis of the generator potential. In the dark, sodium channels in the outer segments of the photoreceptors are kept open by cyclic GMP. So there is a steady current flow from inner to the outer segments of the rods. The steady current also flows in the synaptic ending of the photoreceptors. So when light falls, it leads to the increased conversion of cyclic GMP into 5-GMP, resulting in the closing of sodium channels. And so that occurs because of the decrease in the concentration of cyclic GMP. So this decreases current flow and resulting hyperpolarization, which generates local graded potential, that is generator potential in the synaptic terminal of the photoreceptors. Note that in dark, retinine 1 in rhodopsin is present in Levens's configuration. You can see when rhodopsin, when the light falls on it, it gets converted into bathmorhodopsin, lumirhodopsin, metorhodopsin 1, 2, then gets converted into transretinal and scotopsin. And when in case of dark, the scotopsin is going to bind to the Levens's retinal, which is activated from transretinal, converted into transretinal to form rhodopsin. So here you can see in the disc, when light falls on cis-retinine from rhodopsin, it gets converted into transretinine, and it gets activated. It is going to activate and help in the production of generator potential in the rod or cone. Development of receptor potential or phototransduction. It is a local graded potential generated in the ganglia cells only. The sequence of events involved in the phototransduction of rods and cones are when light stimulus occurs, there will be structural changes in the retinine 1 of rhodopsin. That is, Leven's retinine gets converted into all transretinine, which leads to conformational change in the opsin, which causes activation of transducin, that is G protein, that is GT1 in rods and GT2 in case of cones. Alpha sub, because of which alpha subunit separates from the G protein, and there will be activation of the phosphodiesterase, which converts cyclic GMP into 5-GMP. So which causes decreased intracellular concentration of cyclic GMP, which leads to closure of sodium channels, which leads to hyperpolarization of the receptor, that is decreased release of synaptic neurotransmitter glutamate, and so which causes the disinhibition of bipolar cell through interneurons, and thus an action potential develops in the ganglion cells. So this is the mechanism which occurs in case of DARC, where the high levels of cyclic AMP levels, they keep sodium channels open, sodium enters in, and which causes depolarization, helps in the release of glutamate neurotransmitter. In case when the light falls, so there will be production of the transducin, which causes the decreased levels of cyclic GMP, so which causes the closure of sodium ion. So sodium levels decreases, which is going to lead to hyperpolarization, and that leads to decrease in the neurotransmitter glutamate, and so these are going to inhibit the interneuron, and interneurons in turn excites the bipolar cells. Dark adaptation. When a person enters in a dark room from brightly lit outdoor, at first he sees nothing, but gradually he gets adapted to the dark. The reason is, in bright light, vision was only due to the cones. Cones have high threshold for stimulation. So when the person enters dark room, the low intensity light that is available in the dark is unable to stimulate cones. But visual perception improves quickly at first and more slowly thereafter. So this decrease in the visual threshold and increase in the sensitivity of eye to the light is called as dark adaptation. So here you can see the gradually there will be a decrease in the threshold for the cones, which appears first. Okay, so then the rods are going to be adapted, and gradually threshold for activation of the rod is also going to increase. So that increases the visual acuity in the dark, so which takes around 20 to 25 minutes. So you can see there are two components, fast response and slow response. Fast response is the first drop in the visual threshold and is rapid due to the dark adaptation of the cones, and this is called as paradoxical adaptation of cones, and its duration is 4 to 5 minutes. Slow response, further drop in the visual threshold, occurs over a period of 20 to 25 minutes due to the dark adaptation of the rods. Time of dark adaptation depends on the time taken for resynthesis of rhodopsin. Other important changes that occur in the dark adaptation are there will be dilatation of the pupil, that is mydriasis, and there will be change of photoreceptors sensitivity from cones to the rods, and this is also time required for the resynthesis of rhodopsin occurs when the activated rhodopsin is inactivated by enzyme rhodopsin kinase, thereby reversal of all these reactions occur. So this is in brief about visual process. Thank you.